Photorespiration and mechanism. 1. What is photorespiration? Photorespiration is a light-dependent respiratory process in green cells where oxygen is used, and carbon dioxide is released without producing ATP. 2. What are the requirements and products of photorespiration? It requires oxygen and produces carbon dioxide and water. 3. Does photorespiration produce ATP? No. ATP is not produced during photorespiration. 4. What triggers RuBisCO to combine with oxygen instead of carbon dioxide? When CO levels inside the leaf drop to around 50 ppm, RuBisCO combines with oxygen instead of carbon dioxide. 5. What are the initial products of RuBisCO's reaction with oxygen? Phosphoglycerate 3C and phosphoglycolate 2C. 6. What happens to phosphoglycolate in photorespiration? Phosphoglycolate is converted to glycolate by removing its phosphate group. 7. Where is glycolate transported during photorespiration? Glycolate is transported to peroxisomes. 8. What happens to glycolate in the peroxisomes? It is converted to glyoxylate and then to glycine. 9. Where is glycine converted to serine during photorespiration? Glycine is converted to serine in the mitochondria. 10. What is released when glycine is converted to serine? Carbon dioxide, CO, is released. Disadvantages and evolution. 11. Why is photorespiration disadvantageous to plants? It costs energy and results in a net loss of cofuxation, reducing photosynthesis efficiency by 25%. 12. Is photorespiration essential for plants? No. Photorespiration is not essential, as plants can grow without it if chemically inhibited. 13. Why does RuBisCO bind both oxygen and carbon dioxide? RuBisCO evolved to bind both gases due to its early evolution when the atmosphere had no oxygen. 14. When did photorespiration likely begin? Photorespiration began when oxygen accumulated in the atmosphere. Effect of temperature and gas concentrations. 15. What is the dual activity of RuBisCO? RuBisCO acts as both a carboxylase and an oxygenase. 16. What happens to the O2 by CO2 ratio when stomata close? The oxygen concentration increases relative to CO2 inside the leaf. 17. Why does photorespiration increase at higher temperatures? Higher temperatures reduce CO2 solubility, favoring RuBisCO's oxygenase activity. See photosynthesis. 18. What adaptation do C4 plants have to avoid photorespiration? C plants have a separate pathway called the Hatch-Slack cycle. 19. What is the primary enzyme involved in the C4 pathway? Phosphenol pyruvate carboxylase. 20. What is the first product of CO2 fixation in C4 plants? Oxaloacetate, a 4-carbon compound. 21. In which cells does the C4 pathway start? It starts in the mesophyll cells. 22. How is malate formed in the C4 pathway? Oxaloacetate is converted to malate using NADH. 23. Where is malate transported in C4 plants? Malate is transported to the chloroplasts of bundle sheath cells. 24. What happens to malate in the bundle sheath cells? Malate is converted to pyruvate with the release of CO2. 25. How does the C4 pathway help avoid photorespiration? It increases CO2 concentration in the bundle sheath cells, ensuring RuBisCO participates in the Calvin cycle. 26. What happens to pyruvate in the C4 pathway? Pyruvate returns to mesophyll cells to regenerate phosphenol pyruvate, consuming 1 ATP. 27. Name examples of C4 plants. Maize, sugarcane, sorghum.